Okay, so we're going to go over lab report 5. And I was going to jump right in. However, if anybody has any questions, we can go ahead and address those before we start. Let's see.
My second the aseptic technique lab report still hasn't been, this is too old, by the way, but my aseptic technique report still hasn't been graded.
Yes, I'm working on those. I'm sorry,
Okay.
I am. Yeah. I'm working on those, and so hopefully by tomorrow I'll be finished with those. But I mean, and yeah, yeah, you should have the grade by tomorrow.
Okay, thank you.
No problem. Anything else? Okay, so let me go ahead and get started. And actually what this lab exercise does is it explores techniques that allows us to do attain pure cultures. So the first thing is to know what a pure culture is. And this question has already been asked in previous lab exercises. A pure culture is a bacterial culture in which all of the cells in the culture belong to the same genus, species, and strain because they all arose from the same progenitor cell. So they all came from one cell. At least that's the theory. So it's a population of cells derived from a single cell progenitor. That single cell progenitor is called a CFU, or colony forming unit. And so it is not really called a cell, but a colony forming unit. And that is to acknowledge the fact that it could be two cells and not one. Regardless, a pure culture is assumed to contain organisms that belong to the same genus, species, and strain. So they're all identical because they're clones of one or two cells. So that's the first thing. So oftentimes when it comes to identifying organisms, obviously we need a pure culture. In order to study organisms, pure cultures are again needed. Or to keep stock cultures of organisms, we need to make sure there's only one organism per culture. So in order to create and maintain pure cultures, we need to follow aseptic techniques that will minimize the potential of contamination. So one of the techniques to attain a pure culture is to transfer or to work with a single colony. So when you have an individual colony, that colony is considered a pure culture. By definition, a colony came from one cell, one or two cells, so every single cell in that population is identical. So the easiest way to create a pure culture is to attain isolated colonies, and then from those isolated colonies, pick one and transfer it to a nutrient broth, for example, or a nutrient agar slant, or to another plate. And once growth is attained, then you have a pure culture. So, for example, to make a pure culture of that organism in that colony, all you would do is to transfer a sample of those cells into a nutrient broth, and after growth, that broth contains a pure culture. Okay. So the key is to be able to obtain isolated colonies. That is the key. All right. So there is a technique called the strict plate method for isolation, which is a quick and cheap means to obtain isolated colonies. So the first thing you need to know is that these to strict for isolation means to use this technique that will give us isolated colonies. So the goal is to get isolated colonies that we can use to create pure cultures. Another similar purpose of this plate would be to separate a mixed culture. So if you have a mixed culture and you make a strip plate and you strip it for isolation, you should be able to see the isolated colonies. that belonging to each of the organisms in that mixed culture, and then you will be able to separate. So this is basically how the technique is done. When you proceed with this technique, you need to follow aseptic techniques. With a sterile loop, you're going to pick a sample of a bacterial growth from either a plate, like they have it here, or from a broth or from a slant. Then you're going to come to the plate, to the new plate, where you're going to streak for isolation, and you're going to streak one side of the plate with close together streaks. Then you're going to flame the loop again to kill whatever organism remains in the loop. And what you're going to do from that point on is to dilute the number of organisms on each subsequent portion. We call them quadrants because there's 4 of those. So in each subsequent quadrant, we're going to begin to dilute the number of organisms. So with the just flamed and cooled loop, which has no bacteria in it, you're going to come to the first quadrant and you're going to drag a few organisms into the next quadrant and then streak close together. So what you're doing is you're spreading some of these organisms into another portion of the plate, essentially diluting. You're going to do the same thing again, flame the loop, let it cool, and then you're going to spread from quadrant 2 into quadrant 3 and spread the organisms. So now you have even less organisms than you had in quadrant 2 in quadrant 3. Do it again, flame, and drag a few organisms from quadrant 3 to quadrant 4 and then spread with loose streaks. So again, you're just grabbing a few of the cells from quadrant 3 into quadrant 4 and begin spreading them across the plate to try to obtain individual cells falling in discrete areas that will give you an isolated colony. Yeah, Joel, you have a question?
So you're saying that we take the loop once for the first quadrant, we take the loop, take the sampled colony, and streak it across quadrant 1. Then we flame it to kill whatever is left on there, and then that's going to give us less in quadrant 2, and then again and again. So in theory, the most isolated colonies will be in quadrant 4.
Yes, correct.
Okay.
Yes, exactly. It may not necessarily be, but yes, typically it is quadrant 4 where you're going to get the isolated colonies. Sometimes what happens is that you get isolated colonies on quadrant 3 and nothing on quadrant 4, probably because you picked such small sample that there were not enough cells to drag all the way to quadrant 4. But yeah, ideally you end up with enough with most isolated colonies in quadrant 4. Exactly. Yeah.
And the flaming happens between each spreading.
Yeah, the purpose of the flaming is to kill the bacteria in the loop. So now you have a completely empty loop with which you can spread some of the cells from one quadrant onto the next.
Okay, thank you.
Yeah, you're welcome. So the technique itself is quick. Once you learn it, it's easy and it is effective. It does give you isolated colonies. You can see here a nice streak for isolation plate. This is the first quadrant right here. You can see how close together the streaks are. Then they would have flamed right there, and then notice how they only went once from quadrant 1 into quadrant 2. Quadrant 2 is a little bit hard to see. This is just looks strange because I don't see quadrant 2. It almost seems as if from here they began going in this direction, which I guess is fine. Then they would have flamed again, and the last quadrant would have just gone in different directions. Now, there are different, slightly different techniques. I've seen it with just 3 quadrants instead of 4, and I've seen it a couple of bit of a different way of doing it. The principle is always the same, regardless of how you do it. The principle or the idea is to divide the plate into sections, put the bacteria in one of the sections, and proceed to drag or dilute fewer and fewer cells from one section to the next until you obtain such few cells that you get isolated colonies. So the idea is the same. Sometimes the technique may vary a little bit. In the lab, when we do it, we use the 4-quadrant method. We tell students to take the plate and divide the bottom into fourths, label the fourth 1, 2, 3, 4, and then begin with the first quadrant. Then flame, go to the second quadrant, flame, go to the third quadrant, and then flame and go to the fourth quadrant. And you almost want to have the fourth quadrant to be the one that is bigger if you can't. That way you have more room to spread. Okay, so purpose of the technique is to obtain isolated colonies. That's basically it. If you kind of obtain colonies, you'll be able to discern whether you have a pure culture. If you don't have a pure culture, you could make a pure culture. So in this example here, notice that this is not a pure culture. You have this big white colony here, and then you have a little bitty colony, which is also white, but even the consistency is a little bit better. It looks more different. It looks creamy. So you know there are two organisms growing, and let's say you want to isolate the big one here, which happens to be E. coli, and grow it in a pure culture. Then using aseptic techniques, you could just, and let's say this plate was obtained with the strict for isolation, which is what it looks like, and you can see the quadrants. You can take that E. coli, put it in a broth, and then you have a pure culture there. Okay, so that's essentially the idea behind these stricken a plate for isolation. I'm going to go ahead and, there we go. I want to take a look at the, at the lab report and see what it talks about, especially this very first exercise. Okay, let's see. So we talked about, all right, this is not as I thought. Give me a second. Okay, let me go down. Okay, there we go. Quadrant strict break method. Perfect. All right. The other methods that we're going to look at are the pour plate and the spread plate, I think. Maybe not. Let's see. Pour plate. Okay, so culture and bacteria, quantification of bacteria. Okay. Let me just refresh my memory here real quick. Okay, all these. Yeah. I'm glad I'm doing this. Okay, perfect. All right, so let's go for quantification of bacteria. And it is here. Let me go back here. Pour plates. Is that how we're going to do it? All right, so quantification of bacteria is going to allow us to separate enough a culture where we will be able to actually count cells. So the idea of this technique is to be able to count the number of organisms per mil of a culture. All right, so there are actually two methods that we could use, two physical methods that we could use. There's another, let me just rephrase that. There are two direct methods we can use. There is one indirect method that we're going to be doing also in the virtual lab. So pour plate method is what we would call a direct method of counting bacteria, and I'll show you why it is called direct. In order to count it, you have to separate it because you have to dilute it. You have to have so few organisms that you can actually count. Keep in mind, most cultures have millions of organisms per mil. You don't want to be counting millions of organisms, but if you could make a diluted version of that sample and count the diluted sample, only a few organisms, all you will need to do then is multiply by the dilution factor, and you'll get the number of organisms in the actual sample. And I'll show you how to do that here in just a second. All right, so the first thing is we need to have our culture that we want to count. And in this case, it says, okay, it's a 2 mil suspension, okay. The results we're going to get will be number of organisms per mil. All right, just telling you the culture, where it's coming from. So the first thing we need to do is we need to dilute a sample, and this exercise is showing you what what we actually do in the lab. So we need to tell students what we're going to do with the supplies. We're going to be working with obviously bacteria, so we need to know where we're going to dispose of things. So we're going to be working with pipettes, and the pipettes will be used to pick up microorganisms. So they have to be put in a disinfectant afterwards. All right, so we're going to make dilution tubes. And what we do is we make what we call tenfold serial dilutions. So we start, let me go back here, with empty sterile test tubes. Obviously, we label them. The diluent is going to be a sterile water. So we're going to be diluting our bacteria with the sterile water. We're going to be using 0.9, at least in this exercise we have here, 0.9 mils of sterile water in each tube. That's the diluent. Yes, go ahead, Joe.
Does that affect the pH, or does it will it affect the pH noticeably to for the bacteria?
To use water?
Yeah, to use water instead of
Instead of broth.
yeah.
Yeah, it is definitely a downside. We use water because it's cheaper,
Okay.
because all these dilutions will end up being thrown away. The organism itself is going to be grown in a medium, like it should.
Okay.
But the dilutions are done in sterile water, typically just to save money. The bacteria will stay in the sterile water for just a few minutes. Probably we probably do lose some because yes, you're going to change pH, you're going to change tonicity. All of a sudden, the bacteria are in a very hypotonic environment. So yes, it affects some cells, but we're dealing with millions. To lose a few is not that big a deal. So yeah, but yes, you're absolutely right. It would definitely affect the bacteria. So here you have the 5 tubes. You can see here the 0.9 mils of sterile water. This is our culture. So this is what we're going to count. We want to know how many bacteria per mil we have in that culture. We're not going to call it bacteria per mil. We're going to call it CFUs, or colony forming units per mil. How many of those are in that bacterial suspension? We're going to be making serial dilutions. So we already have the diluent, 0.9 mils of sterile water. Now we're going to make what we call tenfold serial dilutions, and I'll show you why they're called tenfold. All right, so 0.1 mil of the bacteria will be transferred onto tube number 1, which already had 0.9 mils of water. So we're essentially putting 0.1 mil of the bacteria into what will amount of total of 1 mil of water. So this is the math. To calculate dilutions, you take the volume of the microorganism over the total volume, knowing that the total volume, this right here, is actually volume of microorganism plus volume of water. So in tube number 1, we're going to calculate the dilution. Volume of the microorganism is 0.1 mil. Total volume will be the volume of the microorganism again, 0.1 mil, plus the volume of the water, which is 0.9 mils. Notice that the mils cancel, which is exactly what they're supposed to do, because dilution factors should not have units. So if you add all that, now we have a dilution factor of 0.1. 0.9 plus 0.1 is 1. Okay, now this is the problem. 0.1 over 1. In a fraction, you don't want a decimal. So this number really is kind of weird. We really have to clean it out a little bit. So we want to get rid of that decimal. in order to do that, if you multiply numerator and denominator by the same number, nothing changes. So the way to get rid of that decimal is to multiply by 10. So now you would have 1. So the only reason why we're doing this is to get rid of the decimal. We're not changing the number. The numbers, the end result will be the same. This, by the way, is the answer. But the way in which it's expressed is not correct mathematically. So we need to change it to something that looks cleaner and it's mathematically correct. So we multiply both numerator and denominator by 10, and now we're going to have 1 over 10. So tube number 1 is a 1 to 10 dilution of the original culture. You can express 1 to 10 by putting a 1 over a 10, or 0.1 over 1 is a 1 to 10 also. But again, mathematically, this doesn't look, so that's why we just change it to a 1 to 10. Okay, so now this is a serial tenfold dilution, so we're going to continue to do the same thing over and over again. So the next step is to transfer 0.1 mil of the bacteria from tube 1 into tube 2, which already had 0.9 mils of water. So the math is exactly the same for tube 2 as it was for tube 1, except one little caveat that we're going to talk about. So we have volume microorganism, which is 0.1 mil. So let me go ahead and put here dilutions. Volume microorganism. I'm just going to put volume microorganism over total volume. Now, this is the deal, though. This is tube number 2. This is not tube number 1. So there's already a dilution, prior dilution. So we always multiply by previous dilution. Okay, so we have 0.1 mils over 0.1 mil plus 0.9 to give us the total volume. So now we have 0.1, all the mils cancel, over 1. We said, yeah, we multiply by 10 to get rid of the decimal. So now we have, again, a 1 to 10. Okay, so this was a 1 to 10 of the original culture into tube number 1. We do another tenfold serial dilution, and we have another 1 to 10. And that 1 to 10 is a 1 to 10 dilution of tube number 1 into tube number 2. But that's not the number we want. The number we want is how much have we diluted the original culture into tube number 2. This is what we call the total dilution. In order to get the total dilution, we have to multiply by the previous dilution. So the 1 to 10 dilution from tube 2 is multiplied by the 1 to 10 dilution from tube 1. And the answer will be 1 to 100. Okay? And we're going to proceed doing this over and over again through all the tubes. So this one was a 1 to 10. This one is a 1 to 100. This one is going to be a 1 to 1,000. The next one, 110,000. The next one, 100,000. And you should be able to do the math. And whether the numbers that we're playing with is 0.9 mils of water to 0.1 mil bacteria, or something different like 99 mils water to 1 mil bacteria, this still gives you the 1 to 10. This is still a 1 to 10 dilution when you plug in the numbers. So what we're doing essentially is we're diluting the sample, okay? So that we have progressively fewer and fewer and fewer cells. After we do that, let's see. Okay, give me just a second. To be a million, okay, one suspension. Okay, yeah, just to show you, I wanted to see what this was here. Okay, just to show you what we've been doing. Let's say that the number of bacteria in our culture is something we know, okay? Well, just whatever, for whatever reason we know. So let's say there's 1 million cells per mil in the first in our culture. When you dilute that million cells, you're taking 0.1 mil out of that, and which is a tenth of the 1 mil that's here present. So you're going to make a 1 to 10 dilution, which means that this tube is going to have a tenth less than the original tube, or just knocked out a zero, so that gives you 10,000. Sorry, 100,000. That was a million. 100,000. Okay. Now you make another 1 to 10 dilution, or 1 to 100 of the original, and that knocks down another zero, and that gives you 10,000 cells. The next one is another 1 to 10 dilution, so it's going to give you 1,000 cells. The next one, another, I'm going to just put, actually, that should be a positive. Okay, 4. So that will be 100, and the next one will be 10. Okay. So, however, this is because we can do this because this is an example, and we know allegedly how many bacteria were in the first culture. So that defeats the purpose of the whole experiment, which is to find out how many bacteria are in the culture anyway. So this is just to show you what we're doing. We're diluting the sample by factors of 10. Now what we need to do is we need to plate these dilutions. And the way we plate the dilutions is we take melted nutrient agar, and we're going to transfer, put here, 0.1 mil of the bacteria in dilution tube 5 onto a plate. 0.1 mil is just a tiny little drop. So remember, this tube has 1 mil, and we're taking a tenth of that and putting it on the plate. So let me just show you. You put that little bitty drop of bacteria on the empty plate, and by the way, this is an empty plate. And then you're going to pour melted and cool agar. So the little drop is going to completely be mixed with the agar and disperse the little cells all through the agar, inside the agar, on top of the agar, all throughout. The little cells will be dispersed throughout that agar. The agar is solidified and incubated. Okay, and then we're going to count the number of bacterial cells that appear on each of the plates. So we've made plates from each one of these dilutions. Okay, and then we're going to pour the melted and cooled agar on top to mix the cells with the agar, disperse them, and then we're going to make them grow by putting them in the incubator. Okay, all right. We're going to go back now and see exactly what we did. Okay, so a couple of things. What we're doing is we're diluting the samples, and then we are taking the cells in the samples, in the diluted samples, we're taking a tenth of those cells and putting them on a plate and throwing them all over the plate with the hopes that one of those dilution factors has so few cells that each one of those cells can go into a discrete part of the plate and grow a colony. So that when you look at the colonies, you can count them and know that that 0.1 mil that was transferred from the dilution, let's say this is the 1 to 1,000 dilution, and we took 0.1 mil from that dilution, and we threw it onto the plate. And we count the number of colonies, which were also called CFUs, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. And we come up with 13 colonies. What this tells you is this 0.1 mil had 13 colonies in it. So now we know how many colonies in a tenth of a 1 to 1,000 dilution. Now all we need to do is begin to work our way backwards and mathematically calculate, multiply by dilution factors, and figure out how many bacteria per mil on the original culture. So that's essentially what we're doing with these exercises. a few things. This is a pour plate, which means when you mix the cells with the melted agar, many of the cells are going to end up within the melted agar. And so you're going to have tiny little bitty cells that are completely within the agar, and then bigger cells that are growing on top of the agar. You count every one of them, okay? And that's the telltale sign of a pour plate, is when you can see colonies inside the agar. All right, so we said we had 13 colonies. Okay, give me a sec. Now, in order to get a statistically significant sample, you need to count a plate that has between 30 and 300 colonies. So, plate number 1, which came from dilution tube number 1, had 10,000 cells. That's too many to count. That one you shouldn't even bother counting. It will be impossible to count too many cells, but it's still. Tube number 2 has 10 times less, which will make it 1,000 cells. Which you're going to say, well, I guess you could count 1,000 cells, but that is statistically too many. Mistakes will be made. So again, too many to count. You want something between 30 and 300. Don't even bother with this plate either. The next plate, plate number 3, that one has 100 cells. You could that's a perfect number. Between 30 and 300, we could definitely count 100 cells. The ones after that, one has 10 cells, one has 1 cell, too few to count. Okay. So typically when you have a series of dilutions, there may only be 1 or 2 plates that have the right number of cells, of colonies that can be counted. So now we know that the 1 in 1,000 dilution has 100 cells. That is 100 cells in 0.1 mil. Clean this out. Which is what was plated. So 100 cells in 0.1 mil. Okay, how many in 10 times more? That will be 1,000 cells per mil. Tube number 2 has 10 times more, so tube number 2 would have 10,000 cells per mil. Tube number 3 has 10 times more, so it would have, what am I at? 1, 2, 10,000, 100,000, yeah, cells per mil. Original culture will have 10 times more, so that would be 1,000,000 cells per mil. Okay. So this is one way to do it. Just okay, how many cells in this plate that came from this tube, and begin to work your way backwards by essentially multiplying by 10. There's a quicker way to do it. Obviously, there's a formula that all you have to do is plug in the numbers, and that gives you the answer. So let me just make sure that I don't have the formula in here somewhere. I don't think I do, but just in case. No, I don't. Okay. All right. Oh, maybe I do. Right here. Okay. So what we're doing is we're calculating CFUs per mil. That should say 30, not 300. I mean, not 20. Okay. All right, let me do these. Okay, this is the formula. CFUs per mil equals number of colonies over volume that was put in the plate times dilution. Okay, so in the example that we had 13 colonies, or CFUs. We plated 0.1 mil. We have to multiply that by the dilution, which I believe was tube number 3, so that was 1 in 1,000. Okay, so at this point, all you have to do is solve the equation. You don't even need a calculator for that. Let me keep going here. This right here is essentially a 1/10. So now we have 13 CFUs over 110 mils times 1 to 1,000. So 13 CFUs over 1 to 10,000. Okay. Let me clean this out a little bit. So CFUs per mil, 13 over, that's the volume plated, times the dilution. Now they just need to be multiplied by each other, these two fractions down here, and that will give us mils continue, and these are CFUs. 10,000. Okay, again, you can take a calculator and just go ahead and do this math right here, or you don't even need a calculator because what you're looking at here is a division of two fractions. This is one fraction, this is the other fraction, and they're being divided by each other. So 13 CFUs over 1 divided by 1 over 10,000. And that essentially gives you CFUs per mil, 13 times 10,000 CFUs per mil. And so you always express it in that scientific notation. So that will be 1.3 times 10 to the 1, 2, 3, 4, and then 5 CFUs per mil. So there's the answer. A quick way to do it would have been, once you get here, then you know that this number just multiplies by the number up here. Okay, so that would have made it 13 times 10,000. Don't forget units, CFU, don't ever forget units, CFUs per mil, or 1.3 times 10 to the minus 5. Oops, not minus 5, sorry. 5, not minus 5. Okay, CFUs per mil, units. Okay, so you should definitely be able to do these. Calculate make these kinds of calculations. All right, so there's giving you the example that we talked about. Okay. All right, now, this is what the pour plate method allows you to do. Now, we do have another method that allows you to do exactly the same thing. It's called the spread plate method. Don't confuse spread, pour, and streak. Okay, a spread is similar to pour. It allows us to separate and count. The only difference between a spread and pour is where the organisms are growing. In the pour plate method, organisms were growing on the agar and in the agar. So if you had the little agar there, organisms were growing inside the agar and on top of the agar. So on and in the agar in the pour plate method. Disadvantage of that is you cannot get to the organisms that are embedded inside the agar. You cannot physically grab them if you wanted to do that. Disadvantage number 2, you're pouring melted hot agar onto your bacteria. If the bacteria is heat sensitive, the heat of the agar may kill it, and you end up with less organisms. Advantage, however, is that it gives you surface area for growth. So you may not need to make as many dilutions because you have the entire volume of the agar to spread your organisms. And yeah, and this is the pour plate method. I just said the word spread, but I meant this is still the pour plate method, okay? Because the spread plate method does not use heat, and you're literally going to spread the organisms on top of the surface of the agar only. So surface only. That's where the organisms will grow. Okay. The advantage of the pour plate method is that all of the organisms are growing on the surface of the agar. You could actually pick one up and make a pure culture if you wanted to. So it allows you to count and separate. Oh, I didn't say that. I'm sorry. So you have to say, be careful with the name. This is a spread plate. See how easy it is to get them mixed up? Okay, so the spread is the one in which you're only spreading them on the surface of the agar. And let me show you how that's done. And I'll repeat all these since I kind of messed it up. The dilutions are the same. You made the same dilutions exactly the same way. As a matter of fact, in the lab, we use exactly the same tool. So we only dilute once, and we use it for pour plates and for spread plates. The difference is, in a spread plate, you're going to be using a plate that already has agar in it. And the little drop, the little 0.1 mil, is going to be placed on top of the agar. And then we take something called a hockey stick, which is a little rod, glass rod, and we spread, hence the name, spread plate, the drop through the surface of the agar. So when those cells, which hopefully they are diluted enough to where you end up with individual cells falling in discrete areas, after incubation, we're going to see colonies on top of the agar only, individual colonies that can be counted. The advantage of the spread plate method is you're not using heat. The plate is the agar is already made, no heat used, so you don't have to worry about heat sensitivity on the part of the bacteria. Another advantage is you could actually pick up the cells because they're all on the top and create a pure culture if you wanted to. In the pour plate, if the colony was embedded inside the agar, there was no way to pick it up. So be able to distinguish pour and spread plates. The function of both plates, it's very similar. It is counting seeds per mil, with the caveat that the spread allows you to also separate and pick up colonies. Okay. So again, distinguish strict plate for isolation, pour plate for quantitation, spread plate for quantitation. Definitely know how to make the calculations, know the advantages and disadvantages of each one of these procedures. Okay, I'm going to go back to the, oh, this is the wrong one, sorry. This is it, isolation microorganisms. Go back to the lab report. Okay, any questions? All right.
Can you repeat that last bit about the pour plate versus the spread plate? Which one does which? One
Yes.
is only for counting, and the other one is for isolating also?
Yes, so spread plate, count and isolate. Pour plate, counting only. If you're lucky, you could isolate, but that's not as a given. That's not necessarily a given, because in the pour plate, and I cannot draw here, but in the pour plate, you are going to have colonies inside the agar that you cannot get a hold of. So if the colony you want is inside the agar, then you cannot get it.
Okay, thank you.
So it's just counting. Um, let's see. So there's plating. Okay, there's this exercise right here in which essentially they're giving you a tenfold serial dilution. I notice that the volumes are a little bit different from what we work with, but the math should be the same. You should end up with workable numbers. Um, this, for example, they're giving you the, give me a sec. Okay, 110,000. Okay, so you have to calculate dilutions. Oh, yeah, okay. Notice also that instead of plating 0.1, we're plating 1 mil, which makes sense because these bottles have 100 mils. So you are plating more because the volume is also more. Okay. So they're giving you the dilution for, okay, give me a second, 1 mil. Oh yeah, they're giving you the dilution right here, which makes a lot of sense because this is 1, 2, 3, 4. Yeah, that. So this right here is the dilution for tube B. Let me take a look at these on my computer because the what is it? Blackboard Collaborate kind of moves things. I believe this should be over here on the side. It should not be next to B because that belongs to B right there. Okay. So I don't know if you can see my little cursor, but everything is being shifted towards the right. It should be shifted to the left. Okay. So all right. So they're essentially asking you to calculate dilution factors, the volume of bacteria that is being transferred from tube A to tube B, from tube B to tube C. That should be all the same. Dilution factors for B and C, knowing that the diluent is 99 mils of water. And so you should come up with dilution factors, which is what the next question asks you, is to give dilution factors for bottles A, B, and C. Okay. All right. Yeah, so 30 to 300 colonies. Needed to multiply by number of, when calculating the number of bacteria, a sample, in a sample, the dilution factor needs to be multiplied. Yeah, go back to the formula. And this is asking you the number in the denominator of the CFUs per mil formula. So the dilution has to be multiplied by what in order to get the number of bacteria per mil. Okay. Okay, any questions about these? Now, the next, yeah, go ahead. You please do A. Could you please do A? Yes, give me just a second. Let me open it up to where I can take a look at what these actual little arrows are going to. Because the PDF is, I'm going to open it up. I cannot open the Word on Blackboard, but let me at least take a look at where the arrows are going. Okay, there we go. Give me a sec. Okay. Yeah, I still think those arrows are off. Okay, that goes to that one, that goes to that one, goes to that one, and that, I don't know what's the third thing there. Okay, so 1, 2, 3, 4. We just have 3. Okay, let me redo it by hand here, and we'll work on 1. It looks like your screen on ours. Okay, yeah, it actually does too on my Word. So yeah, uh, I would probably let me see if I can redo it tonight. Make it look something that actually makes sense because those little arrows don't really. There are a couple there that are going nowhere, and then I'll send out an email. So let me go ahead and do these real quick. So 1, 2, 3, 1. Okay, so just 3 bottles. Okay, so we have, yeah, no problem. Okay, let's see. Okay, so what we have is the test tube, and then we have 3 bottles, A, B, and C. Okay. Each one has 99 mils of water. Now remember, we're going to calculate dilution factors, and the formula was volume bacteria over total volume. So each one of these bottles have 99 mils of the diluent, and we're transferring 1 mil in a serial dilution. Okay, so the first one would be volume of the microorganism is 1 mil. Total volume is 1 mil plus 99 mils. Mils cancel, and that gives you 1 over 100. Right there. Okay. So this one right here is a 1 over 100 dilution. That's not a 1 over 100. There's 120 zeros. Forget that zero. That's a 1 over 100. Okay. Notice you don't have to get rid of any decimals. You just get an answer immediately. The dilution factor from bottle A to bottle B is going to be the same because we're using the same numbers. However, that's not what you want. You want the dilution factor from the original to 2B, to bottle B. So you're going to have to multiply by the previous dilution. Same here is 1 mil in 99 mils. We're working with the same types of serial dilutions, but that 1 mil into 99 mils gives you the dilution factor between B and C. That's not what you want. You want the dilution factor between original and C. So now you're going to have to multiply by the previous dilution. Okay, so essentially you have to just calculate those 3 dilution factors. And notice that in the exercise, some of these are not being given, but it's given to you is a dilution factor of 10,000. So essentially, to get a dilution factor of 10,000, how many mils you have to change? You have to move from A to B. That's 2 100s. 100 here, 100 here. So it's all exactly the same. It's a serial dilution. Does that make sense?
Do you want this in scientific notation with the 10 to the something?
You can. If you want to do that, that's fine. Definitely scientific notation when it comes to, I'm going to redo it, when it comes to CFUs per mil. So what you're saying is, if you want to use, instead of the 1 to 100, if you want to use 10 to the minus 2, you can do that. You can say 10 to 100. How else you can do it? You can say also 1 over 10 to the 2. All those 3 mean exactly the same thing. When it comes to CFUs per mil, yes, definitely use scientific notation. Does that answer your question?
Yes.
Yeah, good deal. No problem. I have a question about quantitating bacteria in a urine sample is needed. Oops, okay, give me just a second. Okay, so a question about quantitating bacteria in a urine sample is needed to establish a UTI. The minimum number of bacteria in the urine that constitutes an infection is 1,000 CFUs per mil of a single organism. with a sample with 32 colonies of a single organism on a plate that received 1 ml of a 1 to 100 dilution. Okay, so essentially what I'm asking you is to calculate CFUs per mil. So, and this is true in a UTI, in a medical lab, a UTI, the way that they do UTIs is they take the urine and they do a dilution with it to calculate CFUs. They want to calculate CFUs per mil. Now they have a special little loop so they can do this without having to make test tubes or anything like that. But it's essentially exactly the same thing that what we're doing, because technically a UTI is not considered a UTI unless there is 1,000 CFUs per mil in the urine of bacteria or more. So to be a UTI, the result that you should obtain would be 1,000 CFUs per mil. Okay? Now, in the patient, you have 32 colonies. So you want to calculate CFUs per mil, which is number of colonies, over volume plated. Okay, let's just say plated times dilution of the tube, of the dilution tube. Okay, so in this case, the sample has 32 colonies. And you can substitute colonies for CFUs of a single organism on a plate that received 1 mil. Okay, volume plated is 1 mil. And the dilution factor is 1 to 100. Just do the math. And if solving these gives you a number greater or equal to 1,000 CFUs per mil, then the patient had a CFU. And if the number that you end up with is less than 1,000 CFUs per mil, then the patient doesn't have a CFU. I mean, sorry, a UTI. Does that answer your question? Excellent. Good deal. Very good. And by the way, the reason why, yeah, that's okay. Go ahead and if you have more questions, go ahead and ask them. The reason why we have to know the CFUs is because when you collect a urine sample, urine is supposed to be sterile. However, in the process of collecting the sample, you cannot do it using aseptic techniques. The patient is going to introduce contaminants in the urine. So without a UTI, a patient will still have bacteria in the urine, not because the urine had bacteria in the bladder, but because when the urine was collected, bacteria was introduced. So you have to differentiate between the contaminants and enough bacteria to constitute an actual UTI with the bacteria in the urine that is in the bladder, if that makes sense. Okay. So, any other questions? Quantitative dilutions. Okay, give me a second. Yeah, we went over that. We just went over that. That is the little picture of the whatever the bottles, and you're supposed to calculate. So on number 3, on question number 3 of quantitative dilutions, you're supposed to calculate the dilution factors for the bottle A, B, and C from the picture. And I did say I'm going to clean up the picture. The picture is confusing. I'm going to go ahead and clean it up. There are things there that are just look like they don't belong anywhere. I mean, they're just weird. So let me clean it up. But you should have information to be able to calculate the dilution factor. Yeah, go ahead and check the recording. Yes, I know you just joined, so that's okay. I will clean up the picture, and you should be able to calculate dilution factors and fill in the blanks on the picture with information given. Okay, but I'll clean out the picture so that we know exactly where the arrows are coming from and all that. Okay, so that's that one. Okay, one more question. Go ahead. Okay, the calibration set the value of absorption to when there is blank bacteria in the medium. Yeah, let me go take a look at that real quick exactly to see what it is. Okay. Sample was used to calibrate the machine. The calibration set the value. Okay, yeah, we'll do that. Okay. All right, so let's talk about these. Remember that we said pour plates and pour and spread plates allows us to count bacteria directly. Okay. And it is directly because we're actually counting the bacteria. Not only that, but what we're doing is we're counting live bacteria because only those live bacteria in the sample are going to give you live colonies. Okay. Now remember the pour plate are working with hot or warm agar, and that could kill some cells if the cells are sensitive. Nonetheless, you're working with live bacteria, and it's a direct method. Another way of calculating number of organisms in a sample is through an indirect method. An indirect method uses a machine called a spectrophotometer. Okay. All right, it's just a spec for short, actually. And this is what the machine is going to do. If you have a sample of bacteria, notice that when you have a broth of bacteria, the broth is cloudy. So what the machine is going to do is it's going to shine light through the sample. And actually what it does is it makes the light go through a prism, and then it separates the light, and you will shine on your sample whichever wavelength you want. But let's just simplify things, and let's say you're going to shine light on your bacteria. You can measure two things. You can measure how much light, you know how much light you're shining on the bacteria, and then you can measure how much light you collect at the other end. The cloudier the bacteria, the less light will be transmitted at the other end. Hopefully that makes sense. Because the cloudier the bacteria, or the cloudier the culture, the more light will be absorbed by the culture, the fewer rays of light will come out of the other end. And at the other end is the little machine that is actually going to be set off count or determine the amount of light. And it will give us a reading as to how much light has came out. You know how much light you put in. Let's see how much light comes out. That gives you a measure of how turbid that sample is. The more growth a tube has, the cloudier it is, the less light is transmitted. So if you had two samples, and one had hardly any bacteria in it, and another one had lots of bacteria in it, the one that has fewer bacteria will be transmitting more light at the other end. So both are shined with the same amount of light. Let me make 4 and 5. But the one that has more bacteria will have a lower transmittance. This one will have higher transmittance. So measuring transmittance of a sample of a culture, will give you an idea of how much bacteria, how cloudy a sample is. Okay. One of the problems with this method, even though it's very, very good, is that it is an indirect method, and you're accounting for dead and live bacteria. A dead bacteria will still absorb light. So you're getting numbers that are a little bit skewed because you're getting numbers for both live and dead bacteria. Okay, now this is the next thing. What we do is instead of getting a value for transmittance, we're going to turn the coin and we're going to say we're going to get a value for absorbance. In other words, instead of measuring how much light is transmitted, we're going to measure how much light is absorbed. So just for the sake of numbers, just to give an example, not that these are the kinds of things that we measure. We don't measure rays of light, but we have 5 rays of light here. 4 are transmitted, which means 1 was absorbed. The lighter the sample, the less the absorbance. The cloudier the sample, the higher the absorbance. And I'll show you why we measure absorbance and not transmittance. Okay. But you could do either one. You could measure absorbance or transmittance. It doesn't matter. You're doing the same thing. Okay. But this is why we measure absorbance and not transmittance. Okay, so here is the sample. And we said the higher the number of bacteria, the higher the turbidity, the lower the transmittance. Okay. On the other hand, if you measure absorbance, then you're going to say the higher the number of bacteria, the higher the turbidity of the sample, the higher the absorbance. So as you have more and more bacteria, have more and more turbidity, the numbers for the absorbance go up, increase. So mentally speaking, it is easier for brains to retain more bacteria, more absorbance. If you work with transmittance, more bacteria, less transmittance. And yes, you can do it, of course, it's the same, but it is easier for our brains to correlate something that has that does not have inverse relationship. This is a direct relationship. The more bacteria, the higher the absorbance. Okay, so that's easier for our brains to deal with than to say the more bacteria, the less the absorbance. Okay, that's why we measure absorbance. But this is exactly the same. So here we're going to have high absorbance. Okay. Now, this is the deal though. A machine is a machine. A machine doesn't know what a value is unless you tell it, okay? So you have to calibrate the machine. So the first thing we do when we start using these specs is we take a tube with just the diluent, whether it be water or medium or whatever, nutrient agar, no bacteria. And we shine the light on it. And we tell the machine this is zero absorbance. We have to tell the machine what zero is. That's called calibrating the machine. Part of the reason why we have to do this is because we oftentimes are not using water, we're using medium that has a yellowish color to it. So the medium will have a color, the liquid will have a color, but it has no bacteria in it. It's completely clear. And we tell the machine, okay, this is zero, no bacteria, zero absorbance. Anything above these is going to be bacteria, a higher absorbance. But you have to calibrate the machine. This is the equivalent in some ways. When you weigh something and you have to zero the machine against the weight of the container. So you have to tell the machine, okay, this is the beaker. I'm going to put the stuff that I'm going to weigh inside the beaker, but I need to zero the machine with the beaker on. This is exactly the same principle. That's what we call the calibration. So that's what that question is about. The calibration sets the value of absorption to what? You should know that when there is what? How many bacteria in the medium? Does that make sense? Does that answer your question? Perfect. Very good. Okay, let me take a look at this again because what we're doing is they're asking you to do absorbance, I think. Let's see, fill in the blank results of the picture. Oh yeah, the optical. Oh, okay, okay, all right. Small details here. All right, absorbance is also called optical density. It's the same thing. Okay, and hopefully the lab would have told you that at one point or another. So absorbance, we don't call it absorbance, God knows why. We call it optical density or OD. Optical density or OD. So that's why the question says, give the value of the optical density. That's absorbance. Absorbance is optical density. Okay, so the chart wants you to put down the number of the, okay, so this is what it's doing. Okay, let me show you what it's doing. This is actually pretty cool. Declaration set value, okay, look at that. All right. Okay, so that particular experiment, what they want you to do is you have three different tubes with three different bacteria, and you're going to begin measuring the optical density or absorbance of each of these cultures at different time intervals. And remember from chapter 4 that when bacteria grow on a tube, they grow through a growth curve of lag phase, log phase, stationary phase, and then death phase. So as you're measuring the absorbance of each one of those bacteria, they may have a different graph for their growth. So one may have a very short lag phase and then begins a very steep log phase and then a stationary phase. Another one may have a very long lag phase and then it's slowly beginning to the log and then the stationary. So as you're measuring, let's say every whatever it is, I don't remember what the value is, but let's say you're measuring every 20 minutes. At first, on the lag phase, the absorbance value, the OD value, will be the same because the bacteria are not growing. That's the lag phase. And then you're going to see the OD begin to increase tremendously, very quickly. As a matter of fact, you should be able to see doubling in numbers per generation time of OD. So the OD will double as the generation doubles in numbers. So that's the log phase. I don't know that when you get into the stationary phase, that's kind of tricky to do with a spectrophotometer. But you should definitely see lag and logarithmic phase. You may see the slow down into the stationary phase also, at least for some bacteria, if they grow fast enough. So that's what that exercise is doing, is using absorbance to plot the growth of three different bacteria over a period of time by just measuring absorbance. And you're supposed to tell which one grows the fastest, which one grows the slowest, based on, again, the time, the absorbance OD value versus time. Okay. And actually you get a graph and everything. Any questions about these?
I have a question. For the at the beginning, you said that the pure cultures will be the same genus, species, and strain.
Yes.
But so and in last chapter in the textbook, we learned that strains come out when, or strains happen during the death phase, like when they start going Game of Thrones on each other.
Yeah, okay. Well, yes, strains happens during the death phase because of mutations. Yes, correct.
Okay, so uh, will does that do they not occur at um they only happen during the death phase, though, like not in like when we're core plating, we're not going to see strains come out.
No.
Okay.
Okay, so well, let me, yeah, yes, no, it won't. Because there aren't, I'm kind of thinking here. There are four different ways in which strains can come out. Actually, there's five, but there's four that we're going to study. One is mutations. The other three are going to require a virus. So unless there's a virus present, there will be no strains being created. So there will be no strains being created in the log phase of the lag or the log phase. Um, there will be on the death phase because of the stress that the population is feeling for survival. So that will be the accumulation of waste may cause mutations, and then that will lead to strains. Um, strains need another way to create strains is with viruses, with plasmids, or if you have different types of bacteria together, they may exchange genetic material and then you get strains. But in a pure culture over a short period of time, no, you're not going to get strains.
Okay, that
Yeah.
was something that Dave and I had wondered last chapter.
Okay. Yeah. No. No. It's over a short period of time, you're not going to get strains. Yeah.
Okay, thank you.
That's not like, yeah. No problem. Good question. Um, yeah. And on chapter 8, we'll go into how strains are made, into the different mechanisms. So yeah. Um, let's see. Now, having said that, in theory, it would be possible because you could have, in theory, an spontaneous mutation. Yeah, you're getting ahead of yourself. And now I'm thinking, in theory, it's possible. The odds are minuscule. Okay, so for a mutation to happen just due to a mistake in in DNA replication, yes. So okay, so let me go back and rephrase what I said. Yes, in theory, yeah, it is possible. In theory, it's possible. In practice, is um, it's unlikely because the odds are minuscule. But in theory, yes, it's possible. And part of the problem with bacteria is that if you have enough of them, then even something that is unlikely could possibly happen. Okay. So yeah. Um, having said that, we don't worry about the strains when we're doing these kinds of exercises. We assume there are not going to be any strains being created because truly the odds are not very good that there will be any. So yes. Okay, there we go. And now I'm happy with my answer. Yes.
Thank you so much.
No problem. Okay, let's see. Oh, yeah, the reviews just distinguish pores and spread plates. We talked about that. Disadvantages of yeah, and when it says each of these methods, I guess I meant pore plate and spread plate and I guess streak, even though it doesn't say there that. Okay, so go back to advantages and disadvantages. We touched on all that. Any other questions? Okay, so this should be not a terribly hard lab, I think. Maybe the math is the worst, but and obviously it's not terribly difficult math, I hope. But try to get it down because it will be part of the practical. So count on dilution factors having to be calculated and CFUs per mil having to be calculated in the practical. Okay. All right. So there are no other questions. We can go ahead and sign off, and we'll meet again. You are very welcome. Yeah, we'll meet again whenever it is that we're scheduled because I have no idea what my schedule is. Thank you. Thank you. Okay, very good. Thursday. Okay, so we'll meet again Thursday, and we'll cover chapter. Oh, we're going to start. Thank you. It's Thursday at 8:30. We're going to start chapter 5, as a matter of fact, which is an easy chapter. Okay, so we'll start chapter 5 on Thursday. Perfect. And it's not too long. I think we may be able to cover the whole thing. Very good. So, yeah, see you Thursday then.
All right, have a good week.
You too. Bye bye.